Hi everyone, welcome back. In this particular video, we're going to start talking about substituted cyclohexanes and specifically monosubstituted cyclohexanes. So these are cyclohexanes which have one substituent on them. Now, I'm using methyl cyclohexane as an example here, but it would apply to any cyclohexane which has one substituent on it. Okay, and we will develop the chair conformations for these monosubstituted cyclohexanes using the methyl cyclohexane as an example. And what we're going to do here is talk about stability of the molecule in the two conformations, which conformation is more stable than the other. So what I have here is a model of a methyl cyclohexane. Okay, so this group here, read that as a CH3 group. I mean, it is big compared to what a CH3 should be, but that's our new carbon, and those are the 3 carbon-hydrogen bonds, okay? So this is a methyl cyclohexane. And as you can notice, right now, this methyl group is in an, what position would you say this is? This is in an axial position right now, okay? Because this bond here is parallel to that axis. So it is occupying an axial position. Now, if I do, and when the cyclohexane flips, right? So this is right now the headrest in my right hand. My left hand is holding the footrest. And as the footrest comes up and becomes the headrest, and the headrest goes down to become the footrest, what you would notice is that the methyl group is now in an equatorial position, okay? Now, the big question that we're trying to answer here is the stability of the two chair conformations the same, or how do they compare? Is one more stable than the other? And what reasons can we cite for that difference in stability? Okay, so we have the methyl in an equatorial position right now. That's equatorial because in this chair conformation, the green bonds are all axial and the yellow bonds are all equatorial. So when we do the chair flip, it will go to an axial position. The methyl is now axial. So let's start here and then let's try to analyze this cyclohexane to see if we can identify anything that would tell about the stability of this cyclohexane. So let's start by doing some Newman projections again, okay? And since it's a cyclohexane, we're going to do Newman projections looking about 2 carbon-carbon bonds simultaneously, although we just have to focus on one end of this molecule. Okay, so let's do a Newman projection like this. So you're trying to look at this carbon-carbon bond here, the one that I'm holding in my left hand, and a Newman projection about the carbon-carbon bond that I'm holding in my right hand. Okay, and we just have to focus on this carbon-carbon bond because that's where the methyl group is. So when we look at this Newman projection, okay, what would you say about the bonds? I would say that all the groups, they look perfectly staggered, okay? So that is good. It's not an eclipse conformation when the methyl group is axial. Do you notice something else in this conformation? Is there a Gauche interaction that we can identify in this conformation? What about this angle here between those two groups? Sorry, it's hard for me to show, but what you can notice is that in this particular conformation, when we do this Newman projection, there is a Gauche interaction here. There is a carbon-containing group here. That's a carbon-carbon containing group. And there's this methyl group here. These are at 60 degrees to each other. So that means there is a Gauche interaction here. And what we know is that Gauche interactions make the molecule less stable. So that's one thing. Another thing that I want to pay attention, or what I want to bring your attention to, is the hydrogens on this methyl group. Look at this area of space here, okay, in the chair conformation. So if you look at the 3 axial groups here, so we have the 2 hydrogens that are axial, and additionally we have the methyl group here. So there is some crowding going on here, okay? And one of these hydrogens, which is axial, with respect to the methyl group, I'm going to say that they are at positions 1 and 3 with respect to each other. What does that mean? So if I start counting from this carbon, the carbon where the methyl group is connected, okay? If I start from here, notice how if this is carbon 1, then we have carbon 2 and carbon 3. So this hydrogen is on carbon 3 relative to this methyl group, the carbon where the methyl group is connected. And the same is true on this end, okay? Because if I start from here, carbon 1, carbon 2, and then carbon 3. So these are also 1, 3. In fact, these two hydrogens are also 1, 3, 1, 2, and 3. So these three axial groups, they are 1, 3 with respect to each other. And the same is true for the axial groups at the bottom here, okay? So when you have groups, like big groups start showing up, now the methyl is definitely big compared to a hydrogen. So when you have bigger groups that start to show up on axial positions, it results in what's called a 1,3 diaxial interaction. And what that means is two groups that are 1 and 3 with respect to each other, they interact with each other. Okay, that's a steric sort of an interaction, like crowding in space to some extent. Okay, so when the methyl group is in an axial position, we could identify two interactions which would make this conformation less stable, or which would make this conformation less stable. Yeah. So there is a Gauche interaction here. Okay, there is a Gauche between this bond, the one that I'm touching with my fingers, with respect to this bond. There's a Gauche there, and we've identified some 1,3 diaxial interactions here. So that's the conformation where the methyl group is axial. Now let's go to the other conformation where the methyl group is equatorial. Okay, so now the methyl group is equatorial. And now let's look at this bond. Let's look along this carbon-carbon bond again. Okay, that's the same bond that we were looking at before. So if you look at that bond, okay, when you look at that bond, I hope you notice that this is still a staggered conformation. Sorry, I'm just trying to orient it. Okay, this is still a staggered conformation. Okay, yes, there we go. So that's still a staggered conformation. And notice how the methyl group and the other group that were Gauche to each other, which is what I'm holding here, they are now anti to each other. So that means we've got staggered plus anti, which is more stable compared to a Gauche. Okay, so the methyl group in an equatorial position seems like it's better than in the axial position. And additionally, notice how the equatorial position is like more roomy actually in some sense. There are no other groups in this region. So there are no 1,3 diaxial type of interactions. Okay, so essentially what we're learning here is that for a cyclohexane, the equatorial position, okay, is more roomy, or substituents, when they are present in equatorial positions, they are stable, they're more stable. Like having a substituent in an equatorial position makes the cyclohexane more stable because one, we avoid the 1,3 diaxial interactions, and two, we avoid any Gauche sort of interactions because this is now anti. And again, when we say anti, we are referring to these two groups. These are anti to each other. I think this is the best way to look at it. Okay, that methyl group and this carbon, they are anti to each other. Everything else is staggered. But when we were in an axial position, those two groups, those same groups, were Gauche to each other. Okay, so that's a Gauche sort of an interaction between the methyl group and the carbon there. Okay, so that is using the model, but now let's go ahead and draw it out, and we're going to fill it in here. And I'm just going to use the black pen to black marker to represent both the axial and the equatorial positions. So if I start with the first cyclohexane here, and we had the methyl group in an axial position, and since this is a ring, remember we can start numbering wherever, but when you name this compound, carbon number 1 is going to be that, because you want to give your substituent the lower numbering. Okay, so let's just say that this is carbon 1. So this is where our methyl group is. Okay, it is in an axial position. I've just arbitrarily put it in an axial position. Okay, so again, that carbon was pointing up, so axial is up. This is up, so axial is up. Axial is up here. Axial is down, and axial is down. And then, since the axial position is up, equatorial is down, axial is down, so equatorial is up. So we can basically complete this, okay? Fill this thing, H and H. So I've completed the chair conformation. And if we start numbering here, remember this is 1, 2, and 3, okay? Or if we go in the other direction, like we did, like a relative numbering, you could have 1, 2, and 3. So when we talk about axial interactions or 1,3 diaxial interactions, we are referring to an interaction between this methyl group and that hydrogen, and the methyl group and this hydrogen. Now, in this picture, they look really far apart, okay? But that's not really the case. They are close enough. And when we go to like more substituted cyclohexanes, we will see that there is a lot of crowding happening over there, okay? So this here represents our 1,3 diaxial interaction. Okay, and then additionally, like we saw in the Fisher projection, if we look about this carbon-carbon bond, okay, this carbon-carbon bond, then there we will see a gauche between this bond and that methyl group. Those are the two groups that are gauche to each other, because if I look at this carbon, and maybe I will just draw out this part of the molecule. So if I look at carbon number 2, let's say, okay, too small, so let's say 2. If I look at carbon number 2, my eye is here. When I look at it in Newman projection, I see that there is a hydrogen going down. Carbon 3 is going to my right, so that's where 3 is. Okay, that's a carbon. The equatorial hydrogen is going to the left. That is what I see when I look up this bond. Okay, I'm looking from down here up that bond. Okay, and then the back carbon, which is carbon number 1. Okay, so this circle represents carbon number 1. That's the back carbon, and on that carbon, I see the methyl group pointing up. So there's the methyl group. I see hydrogen to the left, and I see carbon 2 going down here. Okay, so these are carbons. This is the third carbon. This is the second carbon here. Okay, and what you notice is these two carbons are gauche with respect to each other. So that's the gauche interaction that we were talking about. Now, once we flip, or once this chair flips, you go to the other chair conformation. The methyl group was axial in this conformation. So in this new conformation, headrest becomes footrest. See, so carbon 1 is over there. And the methyl group, which was axial, is now going to be equatorial. So the methyl group is equatorial. And then other than that, the hydrogen here is axial. And then we have a hydrogen here. We have a hydrogen here, axial, 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 axial. equatorial, 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 and equatorial. And based on what we saw, right? So in this, when the metal is in an equatorial position, then we have no Gauche interactions, and there's no 1,3 diaxial interactions. And because of that, this is the more stable chair conformation. Based on that, we can say that any group, if there's only one substituent on a cyclohexane molecule, it is more favored in an equatorial position than it is in the axial position. Okay, so we'll stop right there, and then what we're going to do is in a follow-up video, we'll start talking about di-substituted and like multiple substituted cyclohexanes. Okay, bye.